Hello, listeners, and welcome to episode 1 of The Deep Dive. Today, we're unraveling the Shroud's Indian secret. It's a story of ancient threads, hidden DNA, and a mystery that spans 2,000 years. So, sit back, relax, and plug in those earphones. Let's get into the data.
What if the DNA hidden deep inside Europe's most famous, heavily guarded, holy relic actually belongs to a stray cat and like a weaver from the ancient Indus Valley?
Yeah, it sounds completely absurd when you first hear it.
Right, because when you picture deeply revered historical artifacts, you usually imagine them sitting in a state of total perfect preservation. You picture them inside this sterile vault, locked behind, 6 inches of climate-controlled, bulletproof glass,
Yeah.
just completely isolated from the outside world.
Well, the assumption is always that these objects exist in a vacuum, right? Like they're completely untouched by the messiness of history.
Yeah.
But when you apply modern genomic science to deeply historical textiles, you quickly realize that for the vast majority of their existence, these objects were completely exposed.
They were just out there.
Exactly. They were out in the open. They were moving across continents and operating essentially as biological sponges.
Biological sponges. I love that. So today we are taking a deep dive into the Shroud of Turin. But okay, let's unpack our mission right up front for you listening.
Yes, very important.
We are absolutely not here to rehash the traditional theological debates or, the political controversies surrounding this artifact. We are setting all of that aside.
Right. That's not what the data is about.
Exactly. Today's deep dive is strictly about the findings of a groundbreaking metagenomic analysis. This was led by Dr. Gianni Barcaccia at the University of Padua in Italy.
Yeah, it's a fascinating study.
And the sources we're pulling from today, which includes excerpts from the Genomic Odyssey of the Shroud of Turin, treat this relic strictly as a biological canvas. We are just going to look at the data and explore the mechanics of how that data actually got there.
And honestly, treating it as a biological canvas is the only way to objectively interpret this new data.
Right.
The researchers, they didn't look at the iconic image on the cloth. Instead, they essentially vacuumed the surface.
Which is just wild to think about.
It really is. They extracted dust, pollen, and microscopic fibers straight off the linen. They approached this ancient artifact the exact way a forensic scientist approaches a deeply contaminated crime scene.
Wow.
Yeah, using a technique called metagenomic sequencing.
Okay, let's actually break down what metagenomic sequencing means, because the mechanics of this are crucial to understanding the rest of this study.
Definitely.
Traditional DNA sequencing usually requires a relatively pure sample from a single organism, right?
Right, like a cheek swab or something.
Yeah, exactly. But with metagenomics, researchers take a bulk sample of an environment, in this case, the dust vacuum right from the shroud, and they sequence all the genetic material at once.
All mixed together.
Yeah. To you listening, think of it like taking an entire physical library, shredding every single book into microscopic confetti, mixing it in a giant pile, and then asking a supercomputer to sift through billions of paper scraps just to reconstruct the French poetry.
That is a great analogy.
Thanks. The computer algorithms separate the human DNA from the plant DNA, the bacterial DNA, and, the animal DNA.
And the computational power required to sort that genetic confetti is just immense.
I can imagine.
But once they mapped out the human DNA found in that dust, the data presented a massive geographical anomaly.
Okay, here we go.
So to understand human ancestry in genomics, scientists look at haplogroups. These are genetic lineages, basically specific mutations in the mitochondrial DNA that trace back to deeply localized populations in different regions of the world.
Got it.
Now, the largest group of human DNA found on the cloth belonged to Near Eastern lineages,
Okay.
making up 55.6% of the total human genetic material.
Which, given the historical and religious context surrounding the relic, a heavy presence of Near Eastern DNA is generally what people would expect.
Right, it aligns with the traditional story.
But the second largest genetic group they identified throws a massive curveball into our geographical expectations.
It really does.
Because according to the data, 38.7% of the human DNA found on the shroud belongs to haplogroups typical of the Indian subcontinent.
Almost 40%.
Yeah. Meanwhile, Western European lineages make up less than 5.6% of the DNA.
What's fascinating here is the discrepancy between those numbers. It is staggering.
It makes no sense at first glance.
Exactly.
Yeah.
You have an artifact with a heavily documented, centuries-long history of being displayed, stored, and venerated in Western Europe.
Right.
Yet the Western European genetic footprint is nearly non-existent compared to this massive South Asian signal.
Okay, I have to stop and push back on this finding for a second, because we really need to interrogate how scientists interpret this data.
That's fair.
Is it possible that this 38.7% Indian DNA is just, I don't know, modern contamination?
Like what?
Like, for example, couldn't this simply be the result of a visiting researcher from Mumbai who handled the cloth during an inspection in the 1970s, and maybe they just happened to shed some skin cells or cough near the fabric?
It's a valid question.
How do scientists actually know the South Asian genetic signal is an ancient footprint and not just a modern accident?
Well, that is the fundamental challenge of studying historical DNA. But the researchers actively accounted for it by looking at the chemical degradation of the genetic material.
Oh, interesting.
Yeah. See, DNA is a highly unstable molecule. Over centuries, it decays. Specifically, it undergoes a chemical process called deamination.
Deamination, right.
Where certain nucleotides literally break down and change their chemical structure as they age.
So it looks physically different under a microscope.
Chemically different, yes. Modern DNA, like from a researcher in the 1970s, has long, intact, healthy strands.
Okay.
But ancient DNA is highly fragmented and shows specific damage patterns at the very ends of the strands.
Oh, wow.
So the algorithms analyzing this metagenomic data, they actively scan for those ancient damage patterns.
And what did they find with the Indian DNA?
The Indian DNA haplogroups detected on the cloth show the severe degradation characteristic of ancient genetic material.
That is wild.
It is deeply embedded ancient DNA.
Yeah.
It is definitely not a recent surface addition.
Okay, so that just upends everything. That's like analyzing an ancient English knight's armor and discovering it's completely covered in matcha tea residue. It completely upends our geographical expectations.
It really does.
So if we have chemically ancient Indian DNA comprising nearly 40% of the human genetic material on a cloth housed in Europe, we have to look at the physical mechanics of how it got trapped there in the first place.
Exactly. We have to trace the journey.
And the researchers developed two primary theories, right? And the first requires us to look at the actual manufacturing process. The manufacturing origin theory proposes that the linen yarn itself was produced in the Indus Valley.
Yes. And to wrap our heads around that, we have to examine how ancient textiles were actually constructed.
It wasn't just a machine pressing a button.
Oh, not at all.
Yeah.
Making linen from flax is a highly physical, biologically intensive process. In antiquity, the flax had to be harvested, retted, beaten, and then spun into yarn completely by hand.
Yeah.
And during the hand spinning process, weavers constantly had to wet the fibers to twist them together tightly.
Wait, how did they wet them?
Often they use their own saliva to wet the threads.
Oh, wow, of course.
Yeah. So the skin cells, from their fingers, their sweat, their saliva, and even the ambient dust of the manufacturing floor, it didn't just fall onto the cloth.
It's just twisted into it.
Exactly. It was physically and tightly twisted right into the microscopic cellulose structure of the linen yarn itself.
So the biological material literally becomes a structural component of the fabric.
Precisely.
And, this makes perfect economic sense when you look at ancient trade. Ancient India was a world leader in high-quality textile production.
They absolutely were.
The Roman Empire frequently imported this incredibly fine Indian linen, which they referred to in ancient texts as Hindoyin.
Yes, Hindoyin.
So here's where it gets really interesting. We could literally be looking at the biological remnants of the people who grew and spun a piece of imported Hindoyin linen thousands of years ago.
It's incredible to think about. The DNA is baked into the original factory setting of the material.
I love that phrasing, baked into the factory setting.
But there's a second theory that layers right on top of that manufacturing origin. And that is the trade route theory.
Okay.
Because even if the flax was grown and spun elsewhere, the genomic data paints a picture of the shroud traveling along the Silk Road.
The famous Silk Road.
Right. It was this vast sprawling network of commerce linking the East and West.
And crucially, for you listening, the Silk Road was not some sterile pipeline where goods magically teleported from one empire to another.
Definitely not.
It was a dusty, grueling, highly tactile network of human interaction. This cloth would have been handled, inspected, unrolled, and repacked by countless merchants and traders of Indian descent long before it ever reached the Mediterranean.
Exactly. Every time a textile is unrolled in an ancient bazaar, it acts like a sponge.
Sucking up the environment.
Yes, absorbing the shed skin cells and genetic markers of the exact people bargaining over it.
So if we connect this to the bigger picture,
the genomic data completely dismantles the illusion that the ancient world consisted of isolated, disconnected bubbles. It highlights a deeply interconnected global economy.
Right. Global trade networks existed long before modern shipping.
Absolutely.
Right.
But human fingerprints are really only one part of the equation here.
Oh, right. The non-human data.
Yes. To fully map the journey of this artifact, the researchers analyzed the non-human genetic confetti. The botanical DNA they recovered operates as a fascinating biological timeline.
And the plant traces are fascinating because they completely corroborate the human data.
They line up perfectly.
The researchers found significant traces of cowpea DNA, and the cowpea is an agricultural crop native to India.
Which aligns perfectly with that massive 38.7% Indian human DNA signal.
Exactly. And they also found genetic traces of various plants endemic to the Middle East and East Africa, tracking a very clear geographical movement.
A physical map through the pollen.
Right. But here is the seemingly impossible anachronistic twist in the data. Among all these ancient Old World plants, the researchers also found DNA from maize and potatoes.
Now, the presence of maize and potatoes is critical. It completely changes our understanding of the cloth's timeline of exposure.
Because maize and potatoes are crops native to the Americas. They simply did not exist in Europe, the Middle East, or Asia until well after the 14th century.
Right, post-European contact with the Americas.
So to you, listening, think about the mechanism of what that actually implies. You have an artifact with ancient Middle Eastern and South Asian origins that is also testing positive for post-14th century New World crops.
It proves that the shroud continued to actively absorb its environment across centuries.
How does the plant DNA even get on there, though? Just blowing in the wind?
Mostly, yes. Plant DNA usually ends up on a textile via pollen. And pollen grains are not perfectly smooth spheres.
Oh, they aren't?
No, under a microscope, they're covered in microscopic spikes and hooks.
Oh, like Velcro?
It's exactly like Velcro. They're designed by nature to cling to the legs of bees. So when wind carries pollen into a cathedral or a courtyard where a relic is being displayed, those microscopic hooks latch permanently onto the woven fibers of the linen.
That is incredible.
So the cloth breathed in the ancient pollen of the Indus Valley, and then centuries later, it breathed in the pollen of agricultural products introduced from the Americas.
It is basically a dynamic, constantly recording document of environmental history, like a 2,000-year-old lint roller.
A 2,000-year-old lint roller, yes, exactly.
And the plant DNA is accompanied by an equally broad array of animal DNA, right?
Oh, absolutely.
Yeah.
The study detected genetic traces from domestic cats, dogs, horses, and various livestock.
Again, we have to picture the physical reality of the ancient and medieval world. Artifacts weren't flying first class.
No, they were transported in the back of wooden carts pulled by horses.
Right, and they were displayed in outdoor courtyards where stray cats roamed around and farm dogs barked at the crowds.
Every gust of wind carried the dander and hair of those animals directly into the weave of the fabric.
So all of this environmental absorption, the maize, the horses, the Indian linen weavers, the African plants, it leads directly to a massive scientific roadblock regarding the human DNA.
It really does. The sheer volume of biological material on this cloth creates the ultimate contamination conundrum.
Right, because the obvious question is, if you have an artifact completely saturated in the genetic material of literally everything it ever brushed past, can scientists sift through that noise to isolate the original DNA of the person who was wrapped in the cloth?
Currently, the answer is no.
Really? Just too much noise?
Way too much noise. You have to consider the cumulative handling of the object. It has been touched by thousands of priests holding it up for display.
Right.
It has been kissed and venerated by endless lines of pilgrims over 2 millennia. It has been handled by researchers, scientists, merchants.
Just layer upon layer of DNA.
Exactly. The cloth is saturated with heavily degraded human DNA from thousands of different individuals across multiple centuries.
Wow.
Because of that overwhelming level of ancient and medieval contamination, pinpointing one singular original DNA profile from the initial rafting is scientifically impossible with our current technology.
And this overwhelming contamination brings us to a crucial point of conflict with past scientific analyses, because many people listening will be familiar with the famous 1988 carbon dating of the Shroud.
Yes, the 1988 study is very well known.
Right. And the carbon dating concluded that the cloth was a medieval forgery, originating somewhere between the years 1260 and 1390. For decades, that was the prevailing scientific narrative.
It was the consensus, yes.
But if this is just a medieval European fake whipped up in the 1300s, how do we account for nearly 40% ancient Indian DNA and deep traces of ancient Middle Eastern and East African pollen?
This raises an important question. The new genomic data seriously complicates that carbon dating narrative. And to understand why, we have to look at the mechanics of carbon-14 testing.
Okay, let's get into it.
Carbon dating measures the decay of radioactive carbon isotopes in organic material. But there is a massive vulnerability when testing historical textiles, and that is biofilms.
Biofilms, what are those exactly?
Over centuries, bacteria and fungi colonize the microscopic spaces between the flax fibers.
Okay.
And these microorganisms actively pull fresh carbon-14 from the atmosphere as they grow, creating a biofilm that chemically bonds to the cellulose of the linen.
Oh, wow. So the bacteria are basically painting a fresh coat of new carbon over the ancient material.
That is exactly what's happening.
Well, wait. Why can't scientists just wash the cloth with harsh chemicals to strip the biofilm off before testing the original fibers?
Because the chemical bond between the biofilm and the degraded ancient cellulose is so incredibly strong. Using aggressive solvents to completely remove the modern carbon would dissolve the artifact itself.
Oh, so it would destroy the sample.
Completely.
Yeah.
Furthermore, centuries of repair work, candle smoke, and human skin oils add newer carbon to the matrix. If a cloth is heavily contaminated with newer carbon, the resulting date will heavily skew toward a younger age.
That makes total sense.
So the 1988 date of 1260 to 1390 might merely be reflecting a massive contamination event, perhaps the buildup of medieval biofilms, or a period when it was heavily handled and repaired.
Rather than the origin date of the flax itself.
Exactly. The deep genetic footprint tells a story of the Indus Valley and the Silk Road that predates the medieval European era entirely.
So what does this all mean? Let's definitively state for you, the listener, what the science is actually saying here, parsing out the data from the noise.
It's important to be clear about this.
This metagenomic analysis does not prove that the shroud belonged to Jesus. It does not prove that Jesus had Indian DNA.
Correct.
It simply proves that the physical material of the cloth has a remarkably complex, ancient, and highly globalized history. It proves the relic is not a simple, localized artifact with a neat, single origin story.
The scientific tools are forcing us to reevaluate our assumptions. Metagenomics is opening up an entirely new discipline for studying deeply historical textiles.
It's really a paradigm shift.
It is. And this isn't just an isolated procedure used to scrutinize the Shroud of Turin. This exact same approach is increasingly being applied to other historical artifacts.
Treating them as geographical canvases rather than sacred untouchables.
Yes. A great example from the sources is the tunic of Argenteuil, which is also sometimes referred to as the tunic of Turin.
Right. It's another famous relic considered a witness to the Passion.
Exactly. And scientists approached the tunic of Argenteuil with the exact same impartial biological methodology.
Looking at the dust and the DNA.
Yes. By mapping the DNA on these types of relics, researchers can trace the historical movements of ancient religious veneration. They can track the paths of trade routes, the migrations of pilgrim groups, and shifting environmental biomes.
All entirely independent of anyone's personal beliefs about the object's authenticity.
Precisely. The DNA maps the human behavior surrounding the object.
To make this abstract science a bit more tangible, if you're listening to this right now, I want you to imagine an old family heirloom sitting in your house.
That's a great way to look at it.
Maybe it's a heavy quilt your great-grandmother made, or a really old family Bible passed down through generations. If you were to run a metagenomic analysis on that quilt today, you wouldn't just find your great-grandmother's DNA.
No, you'd find so much more.
We'd find a massive overlapping composite. You'd find the degraded DNA of your grandfather who slept under it, the dander from a family dog from the 1990s, the pollen from a specific pine tree outside a house your family lived in 20 years ago.
Everything leaves a trace.
The genetic makeup of the object is a composite of every single life that ever intersected with it. It's not just a reflection of the person who originally crafted it. It's a physical, biological map of your family's entire journey through time and space.
And applying that concept to the Shroud of Turin contextualizes this new DNA study perfectly. The artifact is a quilt of human history, woven together by thousands of individuals over thousands of years, carrying the dust of multiple continents.
It's beautiful when you think about it that way. So to briefly recap the incredible journey the data has taken us on today, we started by looking at a relic housed tightly in Western Europe, only to find a staggering 38.7% Indian DNA signal hidden deeply in its microscopic fibers.
A huge surprise.
That data led us down the ancient Silk Road trade routes, exploring the physical mechanics of ancient imported Hindoyin linen from the Indus Valley.
Saliva, the sweat.
Exactly. And we unpacked a biological timeline so vast it features everything from ancient Indian cowpeas to American potatoes that locked onto the fibers post-14th century.
Challenging previous carbon dating models.
Right, and revealing an object that continually breathed in its environment.
The sheer volume of data extracted from a few milligrams of dust just forces us to completely rethink the lifespan of historical objects.
It really does.
But, there is one final detail from this study's findings that offers a particularly grounding perspective.
Oh, what's that?
Well, as we discussed earlier, the researchers found the ambient DNA of domestic cats, dogs, and working horses deeply embedded in the fibers.
Right, the everyday animals.
Yes. We spend so much of our time debating the high priests, the emperors, the saints, and the kings who allegedly possessed or guarded these sacred artifacts.
The famous figures of history.
Exactly. But perhaps the most authentic, unfiltered history of humanity's most revered objects isn't actually told by the powerful people who lock them in vaults.
Wow.
The truest history might just be told by the invisible everyday creatures. The stray cats, the farm dogs, the tired horses that silently brushed past these artifacts as they traveled along the dusty, open roads of the ancient world.
What a thought to leave on. Thank you so much for joining us on this deep dive. We hope this look into the genomic data inspires you to keep questioning the hidden, complex histories resting right on the surface of the things we think we know perfectly. We'll catch you next time.